Hi everybody, today is the first lecture of the protein chapter. Let's talk about what proteins are. Obviously they're found in the cells of living things, living organisms. And there's a difference between protein versus fats and carbohydrates. And the difference is that DNA actually dictates what that protein structure or molecule looks like. You just saw Mandy the Manx. I told you my fur babies would make appearances. Sometimes I don't have control over that. They contain carbon, hydrogen, oxygen, and then the major difference between proteins and carbohydrates and fats is the nitrogen component. The structure of proteins, if you want to visualize it, is a large chain that's linked together by peptide bonds. So I don't know if you had them, but in my biochem class we had these kits that you could purchase and you could actually make different molecules. I never was very good at it. Kind of like Legos, I wasn't good at that either. But it sometimes helps to visualize what the structure looks like. And there's a picture for you to see. And that actual sequence, so the linking of the amino acids in the chain, which are linked by the peptide bonds, determines the shape of the protein. And then that shape, in turn, dictates or determines what the function of that protein is. So it's quite straightforward that if you have 2 amino acids, you have a dipeptide, 3, of course, a tripeptide. 4 to 9 gets a little more complicated or difficult to remember. It would be an oligopeptide, and then 10 or more is a polypeptide. It's somewhat similar to how we classify carbohydrates or lipids. As the chains grow longer, the links are longer, there's more amino acids hooked together, they're more complex. And that leads to a different type of shapes or structures. So there's a 3-dimensional shape, which will determine the function in the body, how it acts. And those, for example, would be tendons. And if you think about a tendon, maybe you haven't necessarily seen a human tendon, but a tendon in a chicken, they're usually longer than they are wide. And an erythrocyte, or red blood cell, tends to be discoidal. If you've ever looked at a slide, that's the very definitive shape that characterizes a red blood cell. So we think about protein in terms of nutrients that we digest, or in a nutrient in the components of food that we eat, but it really has a lot more functions in the body than we initially might realize. The structure of protein. So each amino acid has a central carbon, and I've got a little picture or a diagram there for you. There's 4 sites, you can see them. One of them is a hydrogen atom. You have a carboxy acid group, so there's your acid group on the end on the right, and all those acid groups are identical. And you have the amino group, so that's the nitrogen-containing group, which differentiates it from a lipid or a carbohydrate. Those are also identical. Now, the major difference, or the differentiating factor, is the side chain, the R chain, and that makes each amino acid very unique. So really, that's the takeaway from this slide, that the R group is what makes an amino acid unique. Now, we talk about different amino acids in terms of whether they're essential or indispensable. We use those terms interchangeably. Whether they're non-essential or dispensable, and whether they're conditionally or acquired indispensable. So what does that mean? So if they're essential, we can't make them, so you better be ingesting them in the food you eat. If they're non-essential, the body can in fact make them, so we don't necessarily need to take them in from a food source. If they're conditionally indispensable, we may or may not need to take them in. So there are rate limiting reactions that I will briefly show you. And if you have a disease process, perhaps kidney disease or some type of hepatic disease, you may in fact there are certain amino acids that may in fact become indispensable because your body no longer can metabolize them or make them. So this is a chart from your book. These are the essential or indispensable amino acids, and there's 9 of them. Histidine was the last one to be added. Ignore the darker blue bar at the top. I just didn't change the color, and it just kind of provides a contrast. And I don't expect you to memorize these. I mean, unless you recited these every day, I don't think I could do that even. There is, I believe they call it a mnemonic. Private Tim Hall. It's a good you know, if you want to have a conversation at a party, PVT-TMHLL, and that will help you remember what the essential amino acids are. If you choose to do so, it's not required. The conditionally indispensable amino acids and their precursors I've listed for you. There's 5 of them. And keep in mind, depending on the source, the textbook really doesn't go into this the way that I have, but I'm old school, and this is the way I learned them, so this is the way I'm presenting them to you. So again, these may or may not, depending on your circumstance, whether there's a disease process going on, be something that your body can make. And I like this chart because it lays everything out for you. I've got your indispensable or essential amino acids listed for you, the 9, the private Tim Hall, the non-essential, which I just showed you, the conditionally indispensable, which I just showed you, and then the non-essential. So the non-essential ones, the body can make. But I like them that they're all side by side so you can see them. Again, I don't expect you to memorize this, but it's important to know that there are 3 different classifications or 3 different ways of thinking about how we need or utilize amino acids. So what are the functional roles of proteins? We know that, you know, we take protein in the diet, so what happens after that? Well, proteins are in fact enzymes. They serve as catalysts in the body for various reactions. They're messengers. They're chemical messengers, which means they're hormones. That's obviously very important. Structural elements, if you think about that for a minute, I mentioned tendons, contractile proteins, actin and myosin. Think back to your physiology days. Fibrous proteins, collagen, elastin, keratin, and they also regulate the acid-base balance. So they are quite involved in a lot of the body's physiological processes. Fluid balances, proteins create osmotic pressure. So if you have a low albumin, often you'll have a low oncotic pressure. Immunoprotectors, antibodies, are actually proteins, and they're transporters, albumin, transthyretin, which is really prealbumin, the globulins. Those are all proteins. Additional roles of proteins, acute phase responders. What does that mean? Well, if you've ever heard of C-reactive protein, that at one time was measured after a surgery to see if there were any signs or symptoms of infection. Well, anytime you have a surgery, there's trauma to tissues, and the C-reactive protein will be elevated, so they stopped actually looking at that marker after surgery. And those are usually called positive acute phase responders because they go up in response to inflammation, stress, infection. The negative acute phase responders, such as prealbumin and albumin, go down in response to infection, stress, cancers, things of that nature. So you have 2 different types of responders. One goes up, one goes down. But that'll be important later when we talk about albumin related to nutritional status. Other roles, cell adhesion, signaling, receptor storage, vision. There's a number of roles, very, very important in human nutrition and maintaining homeostasis. Also conjugated proteins I failed to mention, glycoproteins, proteoglycans. So that it's very important to have adequate nutritional stores of protein, above and beyond making sure that your skeletal tissue is functioning and looking as buff as you hope it would. And that concludes my initial lecture on protein, and I will see you again soon. Take care now. Bye-bye.